Hello, everyone. My name is Hector Quemada, and I'd like to welcome you to this special webinar hosted by the Gene Convene Global Collaborative at the Foundation for the National Institutes of Health, and also the American Committee of Medical Entomology of the American Society of Tropical Medicine and Hygiene. Today's webinar will describe recently published guidance and recommendations for laboratory containment of arthropods modified with transgenes capable of gene drive. And our speakers today will be Stephanie James of the Gene Convene Global Collaborative at the Foundation for the NIH, Nicole Achee of the Department of Biological Sciences at Notre Dame University, Mark Bennedict from the Division of Parasitic Diseases and Malaria at the Centers for Disease Control and Prevention, and Zach Edelman of the Department of Entomology at Texas A&M University. Now, following Zach's presentation, there'll be an open question and answer session that will be moderated by myself, along with ACME members Ellen Dotson from the Division of Parasitic Diseases and Malaria at the CDC, and Molly Scheel of the Department of Medical and Molecular Genetics at Indiana School of Medicine. So I'd like to begin then by turning the webinar over to Stephanie James, who will be introducing the subject of today's webinar. Stephanie, please go ahead.
Thanks, Hector, and thanks everyone for participating in this special webinar. Gene Convene's mission is to advance best practices and inform decision-making for development of genetic biocontrol technologies to improve public health. Some of the world's most intractable diseases are transmitted by insect or other arthropod vectors. The growing availability of arthropod genome sequence data, coupled with the increasing ability to manipulate the genomes of these species, is resulting in the creation of a widening array of possible genetically modified biocontrol approaches aimed at reducing the numbers of arthropod disease vectors or reducing their ability to transmit pathogens. These types of genetic biocontrol approaches have been recognized by the World Health Organization and other public health leaders to hold promise as new tools for controlling or possibly even eliminating arthropod-borne diseases such as malaria and dengue. The first step in development of new genetic biocontrol tools is research conducted under contained conditions in laboratories or insectaries. Institutional biosafety officials and biosafety committees are increasingly being asked to assess risks and recommend appropriate containment measures for genetically modified arthropods of public health as well as agricultural significance. A few years ago, the Gene Convene Global Collaborative conducted a survey of biosafety professionals among the membership of ABSA International, an association of biosafety and biosecurity practitioners. The majority of respondents felt that the existing guidance on risk and containment of genetically modified, and especially gene drive modified, insects was inadequate, and that institutions needed additional support for making these containment decisions. As a result of that survey, and in our role of trying to promote informed decision-making, Gene Convene turned for help to the American Committee of Medical Entomology, or ACME, a subgroup of the American Society of Tropical Medicine and Hygiene. Almost 2 decades ago, ACME began publishing comprehensive and widely referenced guidelines for safe handling of arthropod vectors of human and animal diseases. We asked ACME to expand those guidelines to include specific guidance for arthropods modified with engineered transgenes capable of gene drive. And fortunately, they accepted. And those additional guidelines were published in the journal Vector-Borne and Zoonotic Diseases earlier this year. Today, we're very pleased to have several members of ACME with us to explain the purpose of these new guidelines and how they can be used to enhance the safety of research on GM and gene drive modified arthropods. Our first speaker will be Nicole Achee to introduce the guidelines.
As Hector already introduced, I am Nicole Achee. I'm at the University of Notre Dame, based in Indiana of the United States. And I'm a research professor whose program focuses on medical entomology. My role in today's webinar is just to give a brief overview of the Addendum Intent, our drafting process, and just the structure overview. And then my colleagues will take it from there to give more detailed insight about the addendum content and some of the recommendations that we provided. So I think it's important to talk just quickly about what is ACME. As Stephanie said, ACME was asked and we accepted the charge to expand the existing arthropod containment guidelines. ACME is, as you've heard, part of the American Society of Tropical Medicine. It is the American Committee of Medical Entomology. And ASTMH, for those of you who may not be familiar, was established in 1903, and it's one of the largest international societies actually for the control and prevention and research and management of tropical infectious diseases. So for ACME, our mission is to promote the study and control of arthropods that directly and indirectly cause diseases or conditions. So human health harm, animal health harm. And our membership is largely composed of medical entomology professionals, but expanding from academia, industry, government, and non-government. So quite a wide perspective, but all charged against medical entomology. And I thought it was also going to be informative just to start off by briefly defining what is gene drive. So as Stephanie said, we are moving into more refined and rigorous and interesting technology towards genetic modification of organisms, gene drive being one of those approaches. According to what we've published and according to some of the references that we've outlined in the addendum, the definitions that we're going to provide today are specifically linked towards the phenomenon of a biased inheritance. So gene drive is essentially creating a biased inheritance towards a prevalence of a genetic element, natural or synthetic, or some specific alternative form of a gene allele, which increases in presence, even in the situation of a fitness cost. In other words, if there's increased mortality, there's still some increased inheritance there. And this leads to preferential increase of that specific genotype that is leading towards a specific phenotype. So just as a quick example, if you are wanting to manipulate the eye color of an arthropod, you can push that eye color gene forward in subsequent generations at a fast rate. And what happens is all the subsequent generations will have those red eye colors. So that genotype will direct a phenotype in a biased inheritance manner. And this can occur very quickly. Considering the methods that are currently under development for gene drive, what we're doing in arthropod-borne disease research in integrating gene drive is looking at how we can either suppress or modify wild arthropod populations that are responsible for vectoring diseases of human health interest. So these particular arthropods capable of gene drive require some special handling and best practices considerations, which is again why the addendum was initiated. Now, as Stephanie mentioned, we do have existing resources for best practices in research with arthropods, with organisms that may be transgenic. And the NIH was involved with many decades ago developing guidelines for the research with recombinant or synthetic nucleic acids, but there's no specific information in that document at the time regarding containment of arthropods. And so this is where ACME was joined in a couple of decades ago to develop those arthropod containment guidelines. And as Stephanie mentioned, they're published, and many of you maybe have already been referencing those. That guideline was always meant to be a living document in order for us to modify that and add to it as needed to maintain its relevance within the scientific community. It is being referenced widely, but we also want to make sure that it doesn't become outdated. So the current addendum is really intended to expand and complement that existing ACG version 3.2, which is published. And our current new recommendations are termed ACL2+. And my colleague, Zach, will speak to this in a little bit here in the webinar. And these ACL2+ recommendations are really meant to address those standardized needs and requirements for proper handling of arthropods capable of gene drive. Now, that includes the best practices for preventing escape. We certainly want to make sure that they are not removed from the environment under which they're being evaluated. and that we foster responsible conduct of research. So as our intention is to disseminate this standardized requirements of best practices in handling, we want to make sure that we're building confidence within the broader community about the research that's being conducted with GeneDrive. And this is helping to foster that, that relationship with our broader community and, of course, our colleagues at our own research institutions. And that's really this last intention, which is to support the health and safety of the laboratory personnel engaged with the day-to-day activities with these arthropods, other members of our particular organizations where this research might be occurring, the general public in the surrounding area, as well as the animal health and environment that may be in the same geographic space as these institutions. So the committee members that were invited to participate in the addendum drafting are shown here, and I will be going through each of these quickly, as Hector may have already introduced most of us. But myself, Nicole, and Zach, who will be speaking later, Mark Bennedict from the CDC, who will also be speaking today, Ellen and Dotson and Molly Dumonchiel, who are moderating. But we also have other committee members that are not speaking or moderating today. That's Monica Julia Noose from the University of Nevada, and Brian Tarimo from the Ifakar Health Institute in Tanzania. We were invited by ACME to draft the addendum, and that was in August of 2020. We had some lead committee members assigned for certain sections of the addendum, but all of us came together as a group and reviewed each of the sections and came to a consensus on the a completed version. We sought an informal consultation of that completed version to subject matter experts that were outside of the committee. And we received those critiques and we updated and revised the draft. And then we circulated that to ACME membership for their feedback. Because again, this is an ACME initiative in partnership with FNIH, but it's really meant to make sure that we are gathering that input from our colleagues and specialists within arthropod-borne disease research, but also other perspectives of those individuals that are concerned with vector-borne diseases in general and how responsible research is conducted, especially with GeneDrive technology. We received those comments back in February of 2021, and then we subsequently finalized the draft, submitted for publication, and it was online as of January of this year. And that is in the journal Vector-Borne Zoonotic Diseases. So the structure of the addendum, this is just a screenshot of the table of contents. We have our intent, which includes the abstract. We have glossary of terms. My colleague, Mark, who's leading right after me, will speak a little bit more about definitions. We have a risk assessment section, which also includes examples. So how do we intend this information to be utilized, and give some examples for those readers who are requiring this document as a resource to guide them on their decision-making. We also will have the recommendations, and Zach will be talking about that in more detail with specific considerations as to your unique scenario at your institution or your research program overall. And then we have our standard disclosures and funding and references. We want to make sure that we're maintaining relevance with current science. As we said, there are arthropod containment guidelines originally sponsored and developed by ACME a couple of decades ago. With this new addendum, we're modifying it to keep up with the technology today with vector-borne disease research. We anticipate that it may become another need for more modification. So certainly we want to make sure it's current. And our focus within the addendum was on public health vectors. So those arthropods that may be linked to vectoring human and or animal diseases that could lead to some pathogenicity. That spans between mosquitoes, flies, and fleas and mice, but we also included arachnids, which are the ticks and the mites. So we're hoping that it has broad relevance for a number of individuals. And these recommendations, the ACL2+, may also be informative and useful for containment of unmodified arthropods. So although our focus is on arthropods capable of GeneDrive, some of these are best practices for unmodified arthropods as well. and arthropods of agricultural importance. So again, it is meant to be a broad resource for a wide spectrum of users. And then we have developed a feedback form. Zach will speak a bit more to this, that we want to hear from those of you in the webinar, but also others in the broader community within our ACME membership, that when you do have a chance, and if you are using the addendum, if you have a chance to read through it and you're not necessarily involved with the research, but you have comments or you are involved with such research and you have specific critiques, we want to hear from you so we do maintain that relevancy. There is that disclaimer I want to state before I turn over to Mark, that the addendum was written under the purview of the subject matter experts and their opinions, and it does not reflect the position of ACME as an organization or of any of our individual member organizations. And that our principles of responsible research really span into the wider public. It's respect for the wider public. So we are hoping through this addendum and the arthropod containment guidelines in general, that we're fostering that engagement with the wider community, public community, in that respect, to give confidence in the research that we perform is safe and beneficial. I have some references for those that may be interested. It's also in the addendum, and my acknowledgements are to the American Society of Tropical Medicine and Hygiene, of course, ACME, my colleagues who were involved with the drafting, but also the membership overall, and FNIH colleagues with us, Stephanie and David, for spearheading the initiative, Jasmine, who's helping, and Hector, working with the webinar technology today and keeping us on track. We also had Filippo Rendazzo, who was serving as a liaison administrative support for the addendum drafting and submission and final publication. And I thank you for your attention, and over to you, Mark.
Thank you, Nicole. That was a very good introduction to what I have to say. And I'm going to be backing up a bit to some of the origins of the arthropod containment guidelines. And I'd like to set the stage for that by creating a vision, if you will, of the environment that existed when the original arthropod containment guidelines were developed. So the American Committee of Medical Entomology was a very active organization, and I was taking on the chairmanship. And it just so happened to be about the time when we were starting the MR4, what was called the Malaria Repository, which still exists to this day and distributes mosquitoes around the world. And at the same time, transgenic mosquitoes were being developed. And in this environment, Kate Altman, I'd say, was a prime driver. She was over the MR4 at NIAID at the time. And she and a couple of other people devised this idea of developing arthropod containment guidelines. And as we began looking around for guidance that already existed, there were three principal sources, I would say, that influenced the scope of advice that we could rely on. One part was from USDA, APHIS, for containment of non-indigenous phytophagous arthropods and their parasitoids and predators. That's not really what we were concerned about, which was vectors of human disease. BMBL was focused on microbes and was a prevailing standard. And then the NIH recombinant DNA guidelines also existed. But when we looked at these and their suitability for guiding us about containment of, say, for example, transgenic mosquitoes, there was a lot that was lacking. And as Nicole has pointed out, there was guidance. So none of these covered specific situations we were concerned about. For example, exotic vectors whose primary hazard is due to accidental escape. It didn't really cover uninfected domestic vectors. There was no really appropriate guidance for transgenic arthropods, which were all the rage in that day and continue to be a very active field of study. And most of them did not recognize that arthropod behavior is not like that of an infectious agent. They have behaviors that are quite different. And depending on the particular one, they may be attracted to light or they may avoid light. They can also easily be carried on people leaving the laboratory. I'll have to say, at the time when the guidance was being developed, not everybody was excited about it. And that's quite understandable. As scientists, researchers, you're usually primarily concerned with getting the research done. And for many people, having any additional guidance was considered as just friction or more overhead to their primary role. So the draft members of the original version, as Nicole has pointed out, came out of ACME. There was a lot of input from the vector biology and the biosafety community, which we had a very good relationship with. ACME members who really, I would say, carried the heavy weight were Walter Tabachnik, Stephen Higgs, and myself. Although people like Mike Terrell were very vocal and strident, and you really, in developing something like this, it's great to have people who have a variety of opinions who are competing in the realm of the logic and the sensibility to make sure that what comes out at the other end is acceptable. So the first draft was distributed to about 30 people for comment. It was then revised. We made a second draft, which was again circulated, and one of the outlets was list servers, which I don't think really exist in that same form anymore. It was revised again and then published in Vector-Borne and Zoonotic Diseases in 2003. So the original arthropod containment guidelines provided principles of risk assessment, containment advice, and they were meant to be advisory guidelines, not rules. But as several people pointed out at the time, guidance often becomes a rule in the absence of judgment. And so many of the people who are responsible for prescribing, say, for containment practices are not subject matter experts. And so often they can be expected to accept guidelines as really being hard and fast rules. So we were careful, and I think this is important, to specify containment objectives and suggest some methods by which they could be accomplished, but not prescribe specific measures. There's several reasons for this. If you get too prescriptive, technology changes, means change, but also the requirements for different arthropods are different. So what may be perfectly suitable, say, for devitalizing one arthropod may not work at all for another. We wanted something, too, that was compatible with the commonly used methods that were already accepted. And we knew that many people were working with arthropods quite safely. So a large part of the activity was determining what were they doing and did they believe it was effective, and adopting what was already in place. We had to be consistent with BMBL. And so we went through BMBL with a fine-tooth comb because we knew it was established as a standard, as were the recombinant DNA guidelines. So we couldn't eliminate any measures recommended by BMBL, but we could add things on top of that if we thought it was necessary. It deliberately did not consider Drosophila melanogaster, which I think was a good call. You can argue about whether it should be included or not, but certainly it complicates development of these guidelines greatly. I would say the one thing that is overarching is unique about the arthropod containment guidelines is that, unlike BMBL, biological containment is a significant factor. So work with say a tropical vector in North Dakota doesn't present the same risks as it does in Miami, Florida, in the event of an escape. Whereas if you're working with a pathogen, it's hazardous regardless of where you're working with it because it has a direct effect on human health. So we adopted the terminology arthropod containment level, ACL, and we gave it designation of ACL 1, 2, 3, and 4 to parallel what is used in BMBL for the biosafety level of microbes. And then I'll also use the term risk, meaning potential for loss or harm. So originally, the risk considerations that we were most concerned about was, are you working with a pathogen at the same time you're working with this arthropod? Is it an infected or infectious arthropod? So that would constrain the containment level to a BL, a biosafety level, and we wanted to try to parallel the arthropod containment levels with the biosafety levels of the pathogens. This second point, whether the arthropod is transgenic, ended up being fairly controversial among some members of the committee. There were some who very strongly believed that this was the early days of transgenic arthropods. We simply didn't have enough information that they posed any particular risk or hazard. And there was no point in elevating them to any containment level beyond just the basic level. The committee at large, though, the consensus was that ACL2 was the appropriate level for transgenic arthropods. In retrospect, I feel like that was the right call, and partly because the requirements of arthropod containment level 2 are not that different from a biosafety level 2 laboratory. So accomplishing ACL2 is not extremely difficult if you're already running a BSL2 lab. So, but that is just a bit of a background. That was a contentious point about whether the transgenic should require any special containment. In the context of biological containment, one of the questions was, could the arthropod be controlled or locally eradicated by traditional methods in the event of escape? And of course, even among mosquitoes, with which I'm most familiar with, some can be trapped quite effectively, others cannot. And fortunately, I know of no situation in which eradication or a treatment of a zone around an insectary has had to be conducted, but that was one of the considerations that was included. Another consideration was the phenotype of the arthropod, and particularly could that phenotype increase the vector competence. So we didn't want someone who was, for whatever reason, developing a selected strain that was highly competent to work with it under the same conditions as they would one that was disabled. So that was another factor. Another factor was whether there were disabled strains. Many of you will be familiar with the biosafety levels that are assigned to pathogens. If a vaccine is available or some kind of a treatment is available for a pathogen, that can affect the biosafety level. So this, as far as I know, has never been used as a containment measure, but that was a potential consideration that one could make. So we came up with a table that will be familiar to many of you who have looked through the arthropod containment guidelines. And across the top, of course, it's assigning a containment level, and overarching in terms of considerations is the arthropod exotic, and could it persist in the location where the work is being conducted if there were an escape? So that was a huge factor, and it still drives much of the advice, as you'll see from Zach's presentation today. And then its infection status is also supreme. So if you're working with a BSL-2 pathogen, you have to contain it at least arthropod containment level 2. If you're working with a tick and a BSL-4 pathogen, you've got to work with it at 4, or if it's a BSL-3 at 3, and so on. We also considered whether there was a vector-borne disease cycling in the environment. And at the lowest level of containment, which is containment level 1, the answer would be no. And this would be containment practices that were suitable for, say, a rather casual high school laboratory where they're doing demonstration projects and things like this. But above that level, it's really irrelevant. And there are other factors that prevail. And then we listed just a summary in the table of some practices, the primary barriers, secondary barriers, and so on. Now, the way this table was used in the original arthropod containment guidelines is a bit different from the way it's used in the gene drive containment. And Zach will do a very good job, I think, of explaining that. So in this particular table, you just tried to find the situation that fit your research activity best, and that identified the level. For almost all research, it's done at level 2 that I'm aware of. Certainly anything to do with transgenic mosquitoes and exotic vectors is done at ACL 2. We were not concerned about what you see in these pictures here, which on the left is a reaction to a kissing bug bite, and on the right is a reaction to Anopheles gambiae. That wasn't our main concern. It wasn't that mosquitoes will escape and be a biting hazard and annoying people in their gardens or on their terrace. Most of the consideration revolved around harm of the arthropod that was indirect due to the pathogens it could transmit. In contrast to microbes or pathogens that have a direct effect on humans and animals. So we were concerned about these indirect effects. We were also concerned about whether the arthropod species was already established. Certainly, it seems obvious that the escape of a mosquito that's already established in the locale, assuming the phenotype is not dramatically different, doesn't pose much of a risk. If it's exotic, could it become temporarily or permanently established in the event of accidental escape? And that continues to drive, I think, a lot of decisions about where people decide to work on particular vectors and under what conditions. If it did happen, would that pose any particular risk? We're not worried about biting particularly. We're worried about pathogens that could be transmitted. So would that pose a different risk profile? In the case of zoonotic diseases, does the animal reservoir exist in the locale? And is the disease circulating? So these were the kind of considerations we made that were a bit different from what you would for a microbe. I think the impact of the arthropod containment guidelines has been very clear, even though occasionally I'll get a complaint from someone who is grousing about some requirement that their biosafety committee is putting on them. They are widely recognized and implemented, even though they have no legally binding force. But in fact, they have been directly cited by some national biosafety regulations. I've seen one particular set from an African country that essentially quotes the arthropod containment guidelines. So they are useful and have been adopted widely. And I think the reasons are several. They're easy to understand and they're logical. They filled a vacuum, as I said earlier, about what was available and what was needed. And they don't impose any requirements beyond what are really typical laboratory practices that cannot be achieved fairly easily. And you can adapt them to new construction or refurbishment of existing facilities. So they have been widely taken up, and you'll hear people refer to ACL2 or ACL3 or whatever all the time. And there's a general knowledge of what you're talking about when you use those terms. So I want to introduce a couple of terms as a preface to the special considerations we made about risk in this context. Nicole has introduced the idea of gene drive, which many of you, because you're participating in this particular forum, are already familiar with. But it is a phenomenon of biased inheritance in which a transgene becomes more abundant. And I'll use two different terms that need to be clearly kept in mind and in contrast to one another. An autonomous gene drive is one where a single genetic unit contains all of the components necessary for its function as a gene drive system. In contrast, self-limiting gene drives cannot pass indefinitely through subsequent generations. Now, they could be autonomous, but they have some mechanism in them that causes them to decrease in frequency eventually. They're also known as self-exhausting drives. There are two other terms that need to be kept in mind. High frequency threshold simply means that the number of arthropods that carry an engineered transgene has to exceed some proportion of the population in order to begin driving, whereas others require only the introduction of a very small number of individuals to theoretically begin the process of the gene drive dominating the population. And as Nicole said, there were two different general means by which the desirable effect is imposed on the population. One is a suppression phenotype, which is just to reduce the numbers, just like insecticides do, or in contrast, a refractory phenotype, reduced bacterial competence. This could be a direct effect of a transgene on a pathogen, something that changes behavior, and so on. So those are the two general approaches that we considered. So in terms of risk, there were several very dominant factors that we had to layer on top of the original arthropod containment guidelines. Very importantly, is the gene drive autonomous? Could it spread in a wild population if in the event of an accidental escape? If it required, for example, crossing strains in a gene drive situation, or whether there were other features that limited the spread of the drive, that of course would pose less risk than autonomous drive. We also considered whether the purpose of the gene drive was population alteration or suppression, and the consequences of a potential escape are different for those. It considers whether the vector capacity has been modified just as the original arthropod containment guidelines did, and are there effects on other life history traits that should be taken into account. It also, I think, uniquely includes precautions against inadvertent transfer of a gene drive to another laboratory. So let's say that you're working in a laboratory creating transgenic mosquitoes, among which are gene drive mosquitoes, and another laboratory requests a non-gene drive strain. Precautions need to be made to ensure that what is transferred to the second laboratory is not contaminated with a gene drive mosquito. So those were special considerations in terms of risk that we considered for the gene drive addendum. And again, these do not counteract anything that was in the original arthropod containment guidelines. This is layered on top of it. So Zach is going to talk next about the specific things that we devised that were considered to be useful measures for implementation on the practical level.
Okay, thank you, Mark. So we're going to get to the nuts and bolts of what is actually different between ACL2 and ACL2 plus in the broad sense. I know if you are a researcher, you probably ask yourself, what do I need to change? What do I need to do different with ACL2 plus? Do I need to make any changes? Do I need to adopt anything new? And if you're a biosafety professional or regulator, you're thinking, is the work that I'm currently overseeing, does it fall under these new guidelines or does it not? And so that's what we're going to talk about in this last section. Let's see if I can. It's not here. Okay. So here is the updated version of this table that Mark showed, and it's listed as table 5 that we have here in the addendum. And I've highlighted what is new in this part of the table, which is an extra category for our risk assessment, which is the potential for sustained gene drive right here. So we've categorized that as none or not applicable or none to low and moderate to high. And then that maxes out at this 2+ category. And so I'm going to run through a few scenarios. I think when we were developing this table and the feedback we got from various folks, there was some confusion as to how to read it, how to interpret it, and how to then make a judgment about containment levels based on this table. So the scenarios I'm going to show you should be a good illustration of this principle of row, 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 right? So we're going based on each row. So I have 3 scenarios to show you. In scenario 1, we're talking about the generation of a transgenic Anopheles gambiae strain, and Mark used the example of North Dakota. That's probably why it's in my mind as well, with no associated pathogen work, right? And so let's go row by row and say, okay, this is no pathogen. So we can say absent, so we're at BSL1. Then the next row, it is exotic, but is inviable or transient in an inhospitable climate. Then we go to the next row, it is transgenic, so then we'll go up to level 2, and no associated gene drive. And so we've done this for all 4 rows, and we can see that the highest level is the containment level that we will adopt. So this work would fall under ACL2, and that's what the current guidelines, the previous version of guidelines would have said, and that does not change with this ACL2+. In our second example, we have a lab in Florida that wants to obtain a transgenic Aedes aegypti strain and challenge it with yellow fever. No gene drive transgene is present. Okay, so for row 1, we can say infection status can be up to BSL3. The species Aedes aegypti is present in Florida already, so this would be considered indigenous. It is transgenic, so that would push us at level 2, and no potential for sustained gene drive, so that's level 1. So we can see here when that strain is received, in the absence of yellow fever virus, it would be held at ACL2, but when those infections are done, that strain would be moved to ACL3. In scenario 3, we can talk about the evaluation of what we consider a highly active gene drive transgene in Culex species in New York. No associated pathogen work. Okay, so no pathogen for row 1, so that puts us at 1. Species in the local environment, well, Culex Quinquefasciatus is indigenous there, so that's no change. Transgenic, this is a transgene strain, so that activates ACL2. And we've estimated the potential for sustained gene drive is moderate to high. So that means this work would fall under ACL2+, right? So we always take the row that has the highest level as the containment for that work. And each row is as analyzed. So you have to analyze all 4 rows and then figure out which one is the highest. And that helps you set the containment level, right? So now I want to talk more about what this means, this potential for sustained gene drive. And Mark made a lot of these points, and I want to emphasize them here because we had a lot of confusion also when we received feedback from our ACME members and from other experts as to what this was actually referring to, right? So when we talk about potential for sustained gene drive in these guidelines, this is referring to what is anticipated to occur outside the laboratory in the environment in the case of an escape or an accidental release. This is not referring to what occurs in the laboratory where you have very controlled conditions and laboratory strains and maybe specific crosses that might result in a gene drive phenotype. Okay. So what is the likelihood that the engineered transgene will establish in the local environment and exhibit sustained gene drive with the accidental release of a small number of individuals? And so some of the more powerful examples of gene drive we've seen are these homing-based gene drives that can't accumulate any resistance, target allele is highly conserved, and the species is abundant outside the facility. Right? That would be one of our highest possible levels and would certainly qualify here. But you're going to have to do a local risk assessment on a case-by-case basis, right, to determine whether you fall into this high-risk category or whether the risk is actually very low of that occurring. Right, so some laboratory-only gene drive approaches, right? If you have a synthetic target site that's not present in the wild population, if you're using split drives that are going to segregate away from each other, other underdominance-based methods, or if your homing site that you're using actually has abundant resistance alleles already. All of these things are barriers to gene drive in the wild, along with the location of where these experiments are conducted, the abundance of the species in the environment. Everything goes together in this case-by-case approach, right? So even a highly efficient CRISPR homing gene drive, right, can be classified as having none to low if it's in an ecologically restrictive location, and that's been the case. So all of this together, there is no shortcut that a specific technology will always trigger a specific regulation. As Mark alluded to, these guidelines are not that prescriptive. You have to actually do the risk assessment based on the technology, the environment, and the local populations to see whether or not that risk is actually considered moderate to high. And of course, the risk tolerance of the institution, of the country where this research is occurring may also affect whether or not a specific technology is considered high risk, moderate risk, low risk. That may be defined locally as well. So once that determination is made, whether that risk is considered none to low, you remain at level 2. So a lot of current gene drive work might still fall under level 2 if it falls into these low risk categories, may also be pushed to level 2+ based on a lot of these other factors. So the second half of that table, which we include, then this refers to containment level, right? So this is based on column. So if you are selected as level 2+, then you use all of the practices that go under 2+, right? If you're selecting level 2, you use the level 2 practices, level 3 practices. And again, these are our recommendations. So the last couple slides I wanted to go over just highlights some of these differences we see. And again, a lot of these things are present in ACL2 laboratories already. In fact, I've heard the term ACL2+ used anecdotally, I think for 20 years, that people have used this term informally, just describing a facility that may exceed the recommendations of a typical ACL2 laboratory, but they don't quite qualify as an ACL3 laboratory. And so we're kind of formalizing what the field, the community has already been using as a term to describe some of these enhancements, all right? And so some of these things Mark alluded to, right? Directional airflow, very important for pathogen transmission, but in the absence of pathogen, may not be important at all if you have crawling insects or insects that actually regulate their flight and orient their flight towards or away from air currents. All right. We have updates about compliance monitoring, so how often you check the facility to make sure it's intact, that there are no cracks, leaks, otherwise. So based on the risk assessment, you might have to do more frequent inspections, elevated security, so we keep our doors locked, and then a lot of tracking, right? Restricting, more restrictive on the number of people that can use the facility to the fewest possible. Pre-validation of procedures. So if you're going to do a specific technique or manipulation where you're moving an arthropod around in a certain way, at ACL2, this doesn't really require pre-validation, but with these highly invasive gene drive transgenes, you want to make sure that what the manipulation you're going to do is not going to result in a release into the room or into the space of any strain. So you should practice it first with a non-gene drive strain to make sure that that procedure works, and that the people that are trained to use it know how to do it. Right. Better segregation. So right now, ACL2 calls for segregation of work into a different dedicated space or subroom or screened-in area only for pathogens. But we want to apply that also to these highly invasive gene-derived transgenes, separating these out from other strains. So right now, if you have 5 different transgenic strains, you can put them all in the same room. But we recommend taking these highly invasive gene-derived transgenes and separating them away from other transgenic strains, from other wild-type strains, to the same way you might segregate an infected arthropod. Then we have, again, training documentation, increases in monitoring, again, based on risk profile, the ability to genotype and associate a specific genotype with a specific strain. Records not only kept, but reviewed. This is something that comes from a lot of USDA quarantine guidelines, where there's a containment director who's responsible for daily operations and training. At a certain scale, this becomes really important to have for these gene-derived transgenes as well. We specify more things regarding emergency planning, how to stop the research activities in case of an emergency. And then Mark alluded to this as well, this idea of validating strains before they're shipped. We had a lot of concern about laboratories that may use gene-derived transgenes, but also use other transgenes and other wild-type strains. If those strains are shipped to other laboratories, if there's contamination that this doesn't escape the laboratory on its own, but an invasion of a non-gene-derived transgene or a wild-type strain in the same laboratory as a gene-derived that's holding these gene-derived transgenes, those could be inadvertently shipped to another laboratory that may not know that they're there, where the containment levels may not be as high because they think they're receiving a wild-type strain or an innocuous transgenic strain, when really they have small numbers of gene-derived individuals. So really ensuring that that strain validation is present, so that laboratories that are sending and receiving can verify what they're getting. Okay, and so it was not exhaustive. I can't take you through every single section, but that's where the questions come in. So we want to emphasize that these are living documents. They belong to the community, and they are a reflection of what the community is already doing. I don't think anything in them represents something new. These were all gleaned from, we asked so many people to tell us what they were currently doing, and we kind of made an amalgamation of what their best practices currently were. So as people's practices change and evolve, the guidelines will evolve to keep up, to let people know what the community is doing. So please provide feedback on what's working, what's not working, and with that, we'll open it up to questions. And thank you everybody for their time.
Hey, thanks, Zach. Thanks so much for your presentation. Thanks to everybody for the clear presentations that they gave to explain the new guidelines. I'm going to move over to the question and answer period now. And I'd just like to, again, remind everyone that you can either put your questions in the chat function, or if you'd like to actually interact more directly with people on the committee, you can use the raise hand function, and we can put you on and you can ask your questions on the mic. I would like to, I think, begin this question, but just throwing this question out to everybody in the audience to try to answer it for yourself, and that is whether there were any aspects of the ACL2+ guidelines that you might have seen, if you've read the guidelines, that might be especially difficult for you to implement in your own situation. So please think about that answer, but I have other questions coming in that I'd like now to turn over the session over to Molly, I guess. You were the first one to come up, so I'm going to turn this over to you.
We have one that just came in. I think maybe Nicole, but I will let you all decide. What was the most challenging part of developing these guidelines in the addendum?
If that's, I think that's for the entire committee, correct, Molly, overall? Yeah. And
Sure.
yeah, we're all probably going to have our own remarks, but I'll just lead off that I think it is, in my opinion, it was ensuring that the recommendations that were being drafted and suggested would not be unattainable as much as possible within these certain facilities that we would expect gene drive arthropods to be handled. So that to me was quite a challenge. We want to make sure we're doing best practices and guiding on that, but we also need to make sure that we're not creating too cumbersome of a recommendation that really would be unrealistic for some institutions to be able to implement. But that's my opinion.
Thank you. Other thoughts?
Yeah, I'll agree with what Nicole said, because we were really trying to walk that tight wire of getting things the right way so that people were doing things in a way we felt was going to be safe, but not closing the research space to, especially to facilities and institutions in developing and disease affected countries that we're certainly cognizant of the cost of constructing these facilities and their operation. We wanted to make sure that we were writing things that would be attainable in those spaces to keep the space open for folks working in disease intimate countries.
Yeah, I agree with Zach's point. I think that was a overriding concern, and in my evaluation, some of the previous guidance actually would have the effect of cutting off the possibility of doing research in some places, and so that doesn't mean that there was a compromise to accomplish that. But as Nicole said, we tried to keep it practical and effective. But to me, I think the most difficult thing, because I was responsible for risk, was just thinking through scenarios and keeping all of the threads straight to make sure that the practices and the considerations were accurate and realistic.
Thanks, everyone. And I think Ellen is taking the next question.
Well, so this was specifically addressed to Mark and Zach. And once we go through this one, I think we should probably go to the people who have their hands raised. But this one is for Mark and Zach to ask, are there accreditation boards for these containment facilities across different regions, say in Africa?
I know
Mark,
of none such. No.
No. Yeah.
Yeah, in many places, no. And so I think that's why we were cognizant of that. And so the way the guidelines for ACL2 read that we recommend that they reach out to experts outside their institutions or countries to provide that same level of review that it would normally get from an institutional biosafety committee that's present in the US. But we feel that is an important step to make sure that there's a lot of different ways of meeting these guidelines. Every circumstance is unique, and so getting that feedback is always helpful.
So Hector, do you want to call on the hands that are raised? You're on mute too.
Sorry about that. Jasmine, can you turn on? Can you see who's on? Has their hand raised? I'm having trouble seeing that.
Oh, Raul Medina is the first one.
Okay.
Yeah, can you hear me?
Yes, we can.
Yeah, I wanted to know if there is data available from which one can estimate the percentage of escapes that quarantine facilities or containment facilities have, and if there is, how reliable do you think those will be, assuming that some people may not report these based on embarrassment or whatever?
Yeah,
Mark or Zach?
Oh no, I was going to say I have trouble hearing you, Raul.
Yeah, I did too.
Oh,
I think the question was whether or not you have records of how many mosquitoes escape from these facilities, and whether or not you think those records are correct. Is that what you said, Raul?
Yes, that's correct, Ellen.
Okay.
So I think the word escape can have a few different meanings in this context. And so can you clarify whether you mean escape from primary containment, but still remaining within the contained facility, or escaping out of the facility entirely?
Let's say the first level, just to be conservative, like escaping from the first containment level.
Yeah, we keep those records and we use them for training so that folks that we can review those and see if there's a specific technique that is more likely to result in the escape of an individual. And then we change how we do that technique and we revisit training for those individuals. Yeah, so those are very useful records, but I don't know that those are available beyond the individual laboratory that keeps them.
Okay, I mean, that's something that perhaps the committee needs to assess, like evaluate or think a little bit, because, you know, like the public perception aspects of containment facilities are tricky when it comes to transgenic issues, because right now it can be criticized as something that was developed for people that wants to work on these things. But then when you want to go into the public arena, it will be interesting to have that data to make people feel like secure, safe. So it's something that perhaps the committee should start discussing as these things may go into the public.
So this wasn't something that we considered for this committee and this report, because those recommendations, those types of reporting are already required by NIH, at least in the US, and other countries may have their own reporting requirements. So if you have a transgenic arthropod that escapes containment and gets out of a facility, that has to be reported to NIH already, and those become public records.
Yeah. But that's outside of the facility. That's not
outside of the facility, yeah.
Yeah.
But if a person is exposed to that mosquito through a bite or whatnot, that even inside the facility, that would have to be reported.
Thank you.
Yeah, my guess is that with regard to monitoring outside facilities, that's pretty uncommon.
Yeah,
I think in some cases it's probably appropriate not to do outdoor monitoring where the climate is so inhospitable. But even in places where it is hospitable, I suspect it's very, very rare.
It's challenging because we have a lot of mosquitoes outside the facility too. They live there all the time, and
Okay, so the next question is Aegypti Cato. I'm sorry, I'm sure I mispronounced your name.
Yeah, that's how.
Oh, you're on mute again. Sorry. Now.
Yeah, I think I'm on mute now.
Yes.
Yeah, I thank the presenters for all that wonderful presentations. I've been finding a problem differentiating between where I would use SL2 and then SL2. Can I say plus? But I'm getting it a little bit more clear. And my question is about now transportation of SL2 material between labs. How do the conditions differ from transportation of SL1 material? Is there really a difference, or should they be transported the same way? I missed out some sessions, that's why I could have missed it, but I would love to hear about that.
Yeah, Mark, you can chime in on this too. Yeah, go ahead.
Well, so one really ACL1 really requires very little. ACL2, which is where many of you would be working with transgenics, the difference between 2 and 2+, in both cases, you'd have to declare that there's genetically modified organisms and so on. The difference, though, is in the lab that ships the material. They need to examine the material to make sure that it does, if it's intended to, contain a drive or does not. But in terms of the transport, what the courier would see, there's no difference. So it's really that if you're shipping though a wild type strain or a non-gene drive transgenic strain, the lab from which it originates need to ensure that it does not contain any gene drive material.
And the next question
Yes.
that's in the chat was asking about whether or not the containment guidelines are published. And Nicole and Molly have both put this exactly at the same time in the chat. So you can see the link to those guidelines for the addendum in the chat. It is published in, oh my God, I can't think of the name.
Vector-borne zoonotic diseases.
Vector-borne zoonotic diseases.
And it's open access as well.
Yes, so anybody can download the PDF. So the next question is, you mentioned that you consider the criteria adopted in BMBL. Do you consider the risk-based criteria used in WHO's 4th edition? I think we used the 5th edition. Is there some, maybe we should ask this person. This is a meeting administrator. I think we use, the 4th edition for the BMBL now is actually in the 6th edition. Okay.
Who was the one that asked the question? Meeting administrator. I'm not sure if.
It must be somebody in our team.
Yeah. Somebody asked the question. Anyway.
I think we use the 6th edition.
Okay.
Is there something specific about the risk-based criteria that we should have considered that this person sees that we didn't use? I think this person specifically referring to the WHO biosafety. Oh, but the BMBL is manual WHO. It says the criteria used in WHO's 4th edition. Okay. Which they did make a point. They were switching much more heavily to a risk-based approach, and I think that's exactly Zach, and Mark, what you were trying to get to. That these guidelines are taking a risk-based approach.
Yes, definitely.
Okay, great. I think a question was asked. If someone could please speculate, what would it cost to convert an existing 2 to a 2+, and would it be difficult? Hard one.
Yeah, I've been involved in renovations and designs of several level 2 facilities, and I'd say both in Africa and in the United States, and I'd say most of what ACL2 Plus proposes are practices, training, documentation, rather than facility modifications. There are a few things that are recommended more strongly in ACL2 Plus, say, for example, with regard to destruction of material and so on, but they don't require generally extra equipment that isn't already available in most laboratories. You just use it in a different way. So I'd say, look, training and writing documentation, monitoring, those things do cost money because they cost time and they cost staff. And so, but I would say my estimation would be 90% of what level 2 plus recommends is not infrastructure and direct purchases of equipment. Zach, what do you think?
Yeah, absolutely. It's much more in how the facility is used, the training, record keeping, security. So maybe you got to change the way your locks work or not. But it's much more in terms of practices and procedures, and not so much in the facility. The facility is almost exactly the same. There were some things at the lower end, that could barely satisfy the ACL2 that probably wouldn't make the cut at ACL2 plus, but a lot of ACL2 facilities would have no problem just slipping right into these with these additional practices.
So people shouldn't be scared.
And I think that's a great opportunity to remind everyone that the feedback form, we would like to hear if there are some unattainable recommendations according to your own institutions, right? And not necessarily have a specific cost associated with that, but just, we would want to make sure that we're hearing if there was something that seemed to be an ACL2 plus that wasn't seamless, if you will, to transition from ACL to ACL2. So I just think we'd like to make sure we have that feedback from those users of the addendum.
Mm-hmm. Yeah, I'd like to follow up Nicole's comment to make the point that from the very beginning, we felt like the best way for the scientific community to ensure it was conducting itself safely was to propose its own guidance, rather than to have it imposed on people who may not be subject matter experts. And in the beginning, we were using the example of the famous Asilomar conference when recombinant DNA work was just first being developed, where the recombinant DNA scientific community came together at Asilomar in California and came up with some guidance, some of which was later relaxed as experience was gained. But I continue to feel that this approach of having professional organizations build a consensus about the way they practice science and keep it safe and acceptable to the public builds confidence, and it makes it more practical for the scientific community to implement.
Okay, there is a question in the chat by Roland Bamu, and he's asking, why is it so important to contain green drive arthropod, infected or not? And then the second question that he asks is if this method would be applied in Africa to fight malaria, will the mosquito be contained? So
yeah, so that's a really great question, right? So our thinking is that until you have permission by the government and the people around your area to release something into the wild, and a lot of that requires regulation, community feedback and permitting and a lot of other things. Until you have that in place, your responsibility as a researcher is to keep it under containment. And these guidelines are there to help you achieve that goal. So if your ultimate goal is to release and to have an effect on malaria, you'll have to follow all the legal pathway to do that and the responsible pathway of engaging with people who live in the area and the government and everything else, and get all the permitting and all the paperwork that says, yes, you are allowed to do that. Prior to that, it is contained.
Yeah.
Let me just add that there are a couple of people that I noticed that are in the audience that might want to, who are tied into the African regulatory community, that might want to comment at this point too. Sorry, Mark, I didn't mean to interrupt you, but please go ahead.
No, I'll just make a brief comment to follow up with Zach's and to tie the question. All of these arthropods are being created to be safer than the wild type. So we don't anticipate that there's any direct harm if they were inadvertently released. However, every aspect of their safety has not been studied exhaustively. It would be necessary before you could say, yes, there's no risk to the environment, human or animal health. So there's the legal side and the community engagement side that Zach has pointed out, but it shouldn't be misconstrued as a general feeling that these are in some way harmful and we need to prevent that harm from occurring. In fact, we think they'll be very beneficial.
Thank you. I see we have a hand raised. I think Raul, is your hand raised?
Can you hear me now?
Yes, thank you.
Yes. It was mentioned that the presence of animal reservoirs was one of the criteria for deciding if one is going to be of a level or another. But with COVID, we learned that sometimes, new pathogens can establish in things that we were not thinking about, like deer, and some people didn't think in dogs and cats. So I was wondering, do you guys think this is something that should be discussed, or is this addressed somehow in the consideration when it's when you are thinking about animal reservoirs, particularly when you're going to a place in which the animal, the pathogen is, or the vector, let's say, is exotic, and perhaps there are not documented reservoirs there, but is there something there that could act as a reservoir? Is this something that is part of the considerations?
Good question, Raul. And I'm not sure that the answer is clear, except just to clarify that even though there are many of these risk considerations that are considerations, there are only a few that definitely push the containment recommendations from one level to another. So I think much of the decision in situations like that are judgments that are made by biosafety committees and are decided upon as whether they are strong, whether there's strong enough evidence or concern to recommend a higher level of containment. And so I think that that's difficult to answer in a very clear way. Certainly, I think it could be a consideration.
One thing I wanted to say is like,
well, Raul, let me address your question for
yes,
you. Yes. To add on what Mark said. So I think that the absence of a reservoir is really hard to prove, as you noted to, but the presence of a known reservoir should certainly influence the risk assessment, and based on the risk tolerance of the risk assessor and the institution, those unknowns might be treated one way, that there could be a reservoir and we can't rule it out, versus we know that there is a good reservoir nearby, so we are going to take precautions.
Yes, I just wanted to contribute with something. When we import natural enemies for biological control, you know, like wasps and things like that, or even herbivores to attack weeds, we usually bring them to the place and then test with what we think could be potential targets or hosts, and we do experiments like in quarantine facilities usually, and then say, okay, these are what we believe could become problems or non-target hosts for this, and we do a series of tests, let's say, and then we are like, okay, it's unlikely because we didn't observe anything in, let's say, 3 years feeding or trying to make this parasitize that, and then that's part of the evaluation. So I was thinking perhaps something like that could be interesting.
Okay, there is another question in the chat. It says, like Zach said, in the health sector, BSL2+ is well established. However, this is probably not so for GMO regulators or in many territories who have only crop experience. Any precedents where the 2+ have been, any precedents where the 2+ have been accepted by them, or do they tend to push for BSL3? Mark, do you want to take this first?
I know. Yeah, I mean, Zach mentioned that he had heard the term ACL2 plus used.
Yeah.
I have not personally experienced people who had an official status recognizing it yet.
Certainly,
But Mark,
BSL2 plus, right, is people talk about the same thing where you have these items. I'm not aware of any specific experiences, but in terms of what, I don't, if you mean the GMO regulator, you mean somebody from the U.S. Federal government or from institutions. So these are be regulated in the absence of interstate transport or an exotic import at the local level in the U.S. So by an institutional biosafety officer and a biosafety committee, rather than at the federal level. And so my experiences there are that the biosafety community has been very happy with getting this additional guidance for these ACL2+, but maybe if we have any biosafety professionals here that want to chime in as well, they can answer.
Mark, you had mentioned in your talk about seeing these guidelines incorporated into some African country regulations. And I was wondering if you could comment a bit on that. That might be relevant to Henny's question.
Right. So that was, if my memory serves me correct, Kenya. And it referred to the original ACL arthropod containment guidelines. And there was language that was very close to a direct quote, shall we say. And so I think the effect of these kinds of things, I think it takes time for them to be absorbed by the biosafety community and actually probably proposed originally by researchers who are proposing research. So I do think it takes a long time. I mean, the first time I became aware of the arthropod containment guidelines being incorporated was when was during a time, well, you know better than me, when African countries are just developing their own regulations about the containment and use of genetically modified organisms. So it takes a long time and it takes the right environment for these things to get uptake.
Thanks, Mark. We have a couple more comments.
Yeah. Go ahead. Well, we have a comment from Karen Logan, and she's saying, I feel we have to be very careful in our language from ACL2 and BSL2. These are very different biosafety setups. And it's a comment, so no question.
Yeah,
Well, I see that Mark, Zach at least gave it a thumbs up, but Mark, you have a response?
They are different. They have different objectives. But yes, that's right. Everything that is recommended at ACL2 would not satisfy BSL2. But you can operate them both at the same time with no conflict.
Thanks. I see just a general question and an invitation for Brian or some of the other participants in our African communities, if they would like to make any comments in general or ask specific questions. They're putting you on the spot, Brian. Dixon, I think you're there too. Are these, I guess in general, are these useful? Are they applicable? What are the challenges? And anything along those lines? Or if not, you can put them in the feedback. I provided the link in your chat too. So if you need a direct link to that chat and come up with ideas and questions later, please add them there.
I'll comment. I think one of the challenges, and we're just beginning to work with gene-drive mosquitoes in our lab, and that is when you think you're running a pretty tight ship already, the idea of tightening it up even further does present a challenge, and it's more work. There's no question about it. It's more record keeping, it's more monitoring, it's more ensuring your procedures are in place. So it's not without cost. There's no question about it.
Well, it looks like if I don't see any more questions in the chat or anybody else raising their hand to ask a question. So with that, then I think it looks like it's time to just to end this webinar. It looks like we've covered all the questions, and I thank everyone on the panel, all the speakers, and of course those people who tuned in to listen to it. Thanks very much also for your questions and help us understand these guidelines better. I want to also then remind people, as we just talked about, that there is a feedback form that can go to the members of this committee that they can benefit from for any questions or any comments that might be helpful in further improving these guidelines. As they mentioned at the beginning, this is an evergreen document. And so I think they'd appreciate very much any feedback they get, especially from those countries that will be particularly concerned about implementing some of these things in the very near future. So with that, I'm going to thank everybody. Thanks all for tuning in, and we'll see you again sometime in the future.
Thank
Very
you.
good. Thank you.
Thank you.
Bye now.
Bye.